Good afternoon, one and all. On behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nashik, I, Mrs. Rashmi Mishal, Assistant Professor and In charge HOD D-Pharm, extend a warm welcome to you all to the second session of Day 4 of the one-week International e-FDP, e-STP on recent advancements in pharmaceuticals, academic research, ethics, and social perspective under the aegis of Internal Quality Assurance Cell. The speaker for this session is Honorable Dr. Krishna Iyer. It is my privilege to introduce this eminent personality to you all. Professor Krishna Iyer is working with Bombay College of Pharmacy, Kalina, Mumbai as Professor of Pharmaceutical Chemistry and in charge principal. After achieving his MPharm degree from University of Mumbai, Dr. Iyer completed his doctoral studies from University of Minnesota, USA. He has also completed 2 postdoctoral fellowships at University of Washington and Warner Lambert Pharm Research. He joined Bombay College of Pharmacy in 1998 and till date he is working with the same organization. He has guided more than 50 MPharm students and about 7 PhD research scholars. His teaching subjects include pharmaceutical chemistry, pharmaceutical biotechnology, basic and advanced biochemistry, basic and advanced drug metabolism, pharmacokinetics. Dr. Iyer's research interests include mechanistic drug metabolism, evaluation of in vitro drug metabolism, development of in vitro enzyme assays for drug metabolizing enzymes, population phenotyping and genotyping, and drug-drug interaction predictions. He has graced editorial boards of national and international journals of repute such as Indian Journal of Pharmaceutical Sciences and Asian Chemistry Letters. Dr. Iyer has to his credit more than 50 research publications in several national and international journals, and he is a recipient of many awards and accolades. To name a few, the James R. Gillette Award for Best Paper in Drug Metabolism, and he has also received Best Paper Award in Pharmacology and Clinical Pharmacology. He has also received grants of more than 2 crores. His H index is 15 and I-10 index is 19 with over 1077 citations. Dr. Krishna Iyer sir is going to deliver his expert talk today on the topic drug metabolism, role in drug discovery and development. I warmly welcome you, sir. I now hand over the session to Dr. Iyer, sir. Thank you.
Thank you. Thank you very much. Hopefully, you can see my presentation on screen. If somebody can confirm whether it is visible.
Yes, sir. Yes, sir.
Yes, sir. Yes, sir. Presentation is visible.
And let me see if I can get a pointer or something. Let me see. Okay, you can see the arrow now?
Yes, sir.
Yes, sir.
Your cursor is visible.
Okay. Thank you very, very much, Dr. M.S. Gosavi, College of Pharmacy, Pharmaceutical Education and Research. And thank you for the kind introduction. And thank you for inviting me this afternoon to talk to this very wonderful gathering of people who are online. I'm going to talk about drug metabolism and how it plays a kind of a crucial role in drug discovery and development. I'm going to start off with this very simple slide of why we tend to do drug discovery and development, or what are the goals of discovery and development. The first and the foremost thing we are after is to come up with a molecule which is safe when administered clinically. Safety basically implies it should not be toxic on its own. It should not have toxic metabolites. It should probably not take part in any drug-drug interactions, and as far as possible, have no undesirable or off-target effects. Okay, given with this is that even though the drug is safe, it still has to have some utility value. So it does have to engage the correct target to give you the desired pharmacological effect, and it should do so at a very reasonable dose which can be administered, which means it needs to have adequate potency. Now, although these two are the main targets of drug development, that is to find a safe and efficacious drug, it is implicit in this understanding that when you give this drug clinically, that it is able to cross all the barriers which are presented to it inside the system to actually go to the target organ or the target moiety, which is a receptor or a enzyme, at sufficient concentrations to engage it, modulate it, and thereby, kind of give you relief from whatever condition you are suffering. Not only this, this effect cannot be like a very short-term effect. Many times it is required that this effect is sustained. And for that sustained effect, you need the drug to persist in the body long enough, which indirectly means it should have a decent half-life. And given that all of these things are possible, it is also important that from a pharmaceutics perspective, it is easy to formulate. It has the right solubility, dissolution, and chemical stability, so as to allow clinical utility. And most of the pharma companies will target oral administration. So in addition to all of this, it should have the correct bioavailability. So our goals, whenever we're doing drug discovery and development, is to kind of achieve as many as possible of these little goals that we have, so that you get a clinically useful drug. And because many of these requirements need to be put in and require a lot of efforts from different groups who are working, and you'll find that the process of drug discovery and development is fairly slow, takes a long time, and many compounds kind of fall by the wayside as we try to build in all of these properties in them. Now, I only talked about one aspect of it, which is, in a sense, a pharmacokinetic perspective. But then you should also realize that the drug has to have adequate, what you call as, overall safety, chemistry, ease of synthesis, practicality in large scale production, etc. All of these things also kind of contribute to selection of a drug candidate. So typically anywhere from 10,000 to even a million compounds are screened to finally get one molecule which seems to have all the clinical correctness, as they say, to put it in a general population. So it takes a lot of time and it takes a lot of money. Okay? And as we go along this process, there could be different reasons for why a drug fails. And one reason why the drug fails is it doesn't have the correct pharmacokinetics. That means either there is problem with absorption, or a problem with the distribution process, or the problem with the elimination process. And these things can kind of put back your timelines. And in many cases, the drug is even the development is stopped because it doesn't have the correct PK parameters. In our talk, we are going to look at one aspect of this problem, which is metabolism and how it can impact, or and how it is important when you do drug discovery and development. Okay, so drug metabolism, from a definition perspective, is a process by which foreign compounds are converted by enzymatic processes to hydrophilic chemical entities that can be easily removed from a system. Okay, and in that regard, this entire process of drug metabolism, along with what is called the immune system, are two systems which kind of defend us from kind of chemicals and foreign organisms. Okay? Our immune system handles generally bacterial infections or viral infections or fungal infections, and kind of takes care of them by mounting an antibody response. But in the same breath, we are also exposed to a lot of chemicals in our everyday life. Some of them, of course, are the drugs that we actually take. But all of these compounds are considered foreign to our system. And therefore, as a defense against these foreign systems, we have over time evolved enzymes which take these foreign compounds, convert them into something water soluble so that we can remove them in the urine or sweat or in our tears, etc. Okay. So it's kind of like a defense system that we have. Okay. Now, it's very interesting that this defense system is there to protect us from foreign compounds, but it doesn't distinguish when you take a drug deliberately. It considers that also as foreign and will start doing the elimination process. So one can look at drug metabolism in a negative fashion, saying that it is kind of against our intentions of giving a drug and getting an effect. Or one can look at it as a friend of yours, and say, if I understand it, then I can develop a drug which will stay long enough in the body before it gets kind of metabolized, so that I can fulfill my role in taking care of the clinical problem at hand. But then the drug after that is removed from the system, so we go back to what is called as homeostasis. Okay? Now, many organs are involved in drug metabolism. In fact, all the tissues in our body have some enzyme which can do drug metabolism. But if you were to kind of look at it in terms of which is the most important organ, liver is the most important organ, which, as far as drug metabolism is concerned, and that is for two reasons. One, liver is a big tissue or a big organ. It is about a kilogram and a half in an average adult. And then, if you look at it on a per gram basis, it has the highest content of enzymes which do drug metabolism. So liver wins on two fronts. One, it has got a large number of enzymes per gram of tissue, and it itself is a very big organ, about a kilogram and a half. Some of the other important organs involved in drug metabolism, kind of in decreasing order, is the small intestine, lung, kidney, skin, brain. And if you look at it, you'll realize that many of these organs are present at the port of entry through different routes. For example, oral route, it is small intestine and liver. If you kind of chemicals come in through your breath, lung is there. If chemicals kind of go past your skin, skin is there to kind of protect you in a sense. Okay, so in a sense, we are well set up to protect ourselves against foreign compounds, and many times these foreign compounds end up to be drugs. But that is something which cannot be helped in a sense. Now, there are several reactions that are involved in drug metabolism, and typically these reactions are grouped into type 1 and type 2 reactions. Type 1 reactions generally are what are called as reactions where a functional group which is slightly polar is introduced, for example, like an hydroxyl group. And type 2 reactions are those where these functional groups, like hydroxyl or amino groups, are taken, and to them is added a larger entity or what we call a conjugate. And this conjugate is generally very polar, and therefore, when these polar entities are added to type 1 metabolites, the product that you get is significantly more water soluble, and it can be removed very easily in the urine. Okay, an example would be something like benzene, where type 1 reaction would convert it into phenol, which is introduction of an hydroxy group, and then after that the hydroxy group is glucuronidated, or a glucuronic acid moiety is added. So this glucuronide conjugate of phenol now is very, very water soluble and removed from the system. Okay, the enzymes which actually do type 1 reactions are broadly listed as cytochrome P450s, flavin monooxygenases, alcohol aldehyde dehydrogenases, esterases, amidases, and epoxide hydrolase. Okay? And the first two of them do predominantly oxidation, that is cytochrome and flavin. Alcohol aldehyde dehydrogenase do both oxidation and reduction. And the bottom three, esterase, amidase, and epoxide hydrolase, usually do a lot of hydrolysis type reactions. Now, the type 2 enzymes are all referred to as transferases, and depending on which group they transfer, the name is given. For example, the enzyme which transfers glucuronic acid is called as glucuronil transferase. The one which transfers a tripeptide called glutathione is called glutathione transferase. An enzyme which transfers a sulfate group is called sulfotransferase. The enzyme which transfers an acetyl group on a nitrogen is called N-acetyl transferase. And then we have a whole bunch of methyl transferases, which transfer the methyl group, sometimes onto a nitrogen, it's called NMT, sometimes onto a sulfur, called as thiomethyltransferase, sometimes to a phenolic OH, called as phenol methyltransferase, or sometimes to a catecholic group, called as catechol O methyltransferase. Again, note all of these are written in plural, indicating it's not a single enzyme but a family of enzymes. Collectively, if you were to look at all of these enzymes, you'll find that in terms of number, we have about 250 to 300 enzymes which do all the drug metabolism that is needed by us in our entire lifetime. Okay, and they are so nicely designed that they are able to kind of accommodate compounds in a very versatile fashion. For example, if P450 cannot handle it, some other enzyme will be able to metabolize it, and so on and so forth. So they work together. They have what is called as broad an overlapping substrate specificity, and therefore they're able to metabolize millions of compounds, even though their number is only around 300. To give you an example of what I'm saying is, the cytochrome P450 system itself is said to metabolize close to 60 to 80% of all the clinically useful drugs. And within that also, there are some specific family members like 3A4, 2D6, which do most of the metabolism. The other members are relatively less contributing to overall drug metabolism. This is what I meant by family of enzymes. These are all cytochrome P450s. They have different names because their amino acid sequences are different and their substrate specificity is slightly different. But collectively, they're able to handle more than 70% of all the drugs which are there in the clinic. Okay. So here is a simple example of how, for example, aspirin gets metabolized in our system. So this is aspirin here. The first reaction that occurs, and to a significant extent, is hydrolysis, where this group is removed to form acetic acid, and what is left with is salicylic acid. Okay. So now salicylic acid itself is excreted in the urine, but it's also converted to all of these metabolites. This is a glucuronide here, where a glucuronide is added on the carboxylic acid group. Sometimes the glucuronide can be added also to the hydroxy of the phenolic OH. Sometimes amino acid glycine is conjugated to the acid. Sometimes it's further oxidized to give you this, catechol type metabolite. So if you look collectively, all of these things are now present in the urine, and they account for kind of the 100% of dose of aspirin that you take. This is a very simple pathway. If you look at a drug called Indinavir, you'll find there are a lot more metabolites. Okay, in this case, about 8 to 9 of them. And in some cases, for example, like propranolol, it increases further. Here, the number of metabolites are close to about 10 or 11. And then something like valdecoxib, for example, in the middle here, there are about 20 to 30 metabolites. So it all depends on what is the chemical entity we are talking about. Some of them are very simple metabolic pathways. In some cases, the metabolism can be fairly complex and involved. Okay. And when you start drug discovery and development, you are not quite sure which type of metabolism you are going to see. Is it going to be simple, 2 or 3 metabolites, or is it going to be very extensive? And understanding this metabolism is very crucial towards the overall development of the drug. Now, given that fact, how do we try and find out how a chemical is metabolized in a system? Okay, there are several what you call as models for studying drug metabolism, and I've listed all of them here. And if you look at it from the top left, which is single enzyme system, to kind of somewhere in the bottom right, which is in vivo studies, the systems are listed in increasing order of complexity. The simplest system are enzyme systems. Then we have what are called as subcellular fractions. So it could be like a cytosol or a microsomal fraction. Then you can also do metabolism with whole liver cells, which are called as hepatocytes. Or you can study metabolism using slices of livers, which are called as tissue slices. Or you can do what is called organ perfusion, in situ perfusion, where you put the drug through the portal supply, allow it to go through the liver, and then collect it on the other side to see what is happening to the drug. And finally, of course, you can do an overall in vivo study where you give a drug, sample the blood at different times and also sample tissues at different times to get to know which metabolites are formed and where all they end up. Of course, in today's day and age, in addition to all of these, what are called experimental tools, we also have what are called as in silico tools or computer-aided tools, which also kind of contribute to many times the design of the actual in vitro experiment. So we have a lot of systems with us in order to study drug metabolism. And as you can kind of see, the simpler the system, it is much easier to do the experiments, but you will not get the full story. And on the other hand, we have a very, kind of a whole system in an in vivo study. But then, because it's a whole animal or a whole human, you'll find analytical difficulties and methodological difficulties. So, depending on what is the question you're trying to answer, you could use a simple system or a very complex system in order to understand what is happening with respect to drug metabolism. Okay. Now, with respect to the information that is typically required during drug discovery and development, there is a list of what are referred to as the experiments that you generally always do. Okay. One is called as metabolic stability. The second is referred to as metabolic profiling. Third is cross-species comparison. Fourth is referred to as reaction phenotyping. And then fifth and sixth are enzyme inhibition and induction. I kind of will go through very quickly as to what these things are and what is it they're trying to answer and what type of information they're trying to generate, so that you get a feel for this process of discovery and development. Okay. So before we go there, very quickly, as I told you, we have different systems at our disposal to do these studies, starting from single enzymes to kind of slices or even in vivo. And each of them has an advantage or disadvantage, and they have very specific utility. Okay, for example, express enzymes are pure systems, but they are also single enzyme systems. You'll have difficulty in integrating with other enzymes. On the other hand, slices are easy to prepare, integrity is maintained, but then because they are slices, sometimes compounds have difficulty in penetrating through all the cells which are there in the slice, and you might get kind of results which may not be reliable all the time. Also, these slices don't have a long life, so you need to do the experiments very quickly before the liver cells simply die off and do not function as they are supposed to do. Okay? So let us start looking at some of these things. Okay, but before we go into the technique, just a quick note of warning. When we do drug discovery and development, we do a lot of studies in animals, and then looking at the data that we generate in animals, finally we give it to humans and generate information in humans. Okay, you have to remember that when we do work with animals, they are different from humans in different ways, and you need to keep that into account when you try to interpret the data that you have obtained in animals, and then kind of look at them as an indicator of what is going to happen in a human. Okay, so some of the things that you remember are that small molecules, small animals tend to do metabolism much faster than larger animals. Okay, so when you compare something like a mouse versus a human, on a gram to gram basis, mouse will do metabolism much faster than a human. Also, some of the enzymes which are present in mice or rats may not be present in humans. Okay, so the enzyme isoforms can differ. And most interestingly, some enzymes are not even present in certain animals. For example, if an enzyme like glucuronidase transferase is not present in what are called as I'm trying to remember. Glucuronidase transferase is not present in cats. Then N-acetyl transferase is not present in canines like dogs and foxes. Similarly, pigs cannot do sulfation, or they do not have sulfotransferases. Okay. Likewise, in humans also, depending on the age that we are talking, neonates do not have glucuronidase transferase. Very old people have low levels of glucuronidase transferase. So all of these things have to be taken into account when you try to interpret the results. Okay, so you realize that it's not very easy to predict human metabolism from animal data, given all of these constraints. But even then, these studies are done in order to get some understanding of what is happening. Okay, so the first reaction that generally most companies will do is what is called as metabolic stability. Okay, the idea here is to kind of see how quickly or how slowly the drug gets metabolized through, in general, what are called as oxidative pathways. Okay, and typically this is done in vitro in microsomes or hepatocytes. Okay, there are some assumptions that we make when we do these studies. The assumption that we make is metabolism is an important determinant of the overall half-life of the drug. And second, that in most cases, metabolism is going to be dominated by oxidation. So what is it that we are trying to look at when we do metabolic stability? Essentially, what we are trying to do is make sure the drug doesn't get metabolized too fast, that is, has a very short half-life, or has got a very long half-life. That means half-life like a day or two. The unwritten rule is that when you do drug discovery and development, you're going to try and look for a drug which has got a half-life of about 6 to 8 hours. And the reason this half-life is targeted is because most companies in the end want to kind of come into the market with a orally available drug, which can be given kind of once a day kind of system. So if I need to give something as a once a day, I have to make sure that in 24 hours, the drug doesn't get totally eliminated. Neither is the half-life of the drug more than 1 day, such that it doesn't, the concentrations don't come down significantly in a day. So if you have a half-life of say 6 to 10 hours, then in maybe 2 or 3 half-lives, which is 24 hours, you'll find that the drug concentrations, if you start with 100, will go to 50, then 25, and then 10 in that time. And that becomes a little more easier to handle in terms of a 1 day dosing or twice a day dosing. Okay. Also, at this point, we don't worry too much about species differences because in most cases, we are not looking at an absolute number. But if you have a group of compounds, you try to do these studies and rank order them in terms of which has the longest and shortest half-life, and then try to pick something in between, which is probably a more reasonable bet as far as the later processes are concerned. In some cases, if we know why half-life is short due to specific metabolic pathways, then this information can be then given to the medicinal chemistry group to tell them, okay, metabolism is occurring at this point. So can you change the functional group there or the group there so that you can slow down metabolism? Okay. One example of that is a drug like this. This is ethyl benzene. This gets metabolized very, very fast. So if I want to kind of slow down the metabolism a little bit, because metabolism most dominantly occurs at this CH2 here to give CHOH, what I do is replace the CH2 here either with a single fluorine or two fluorines. Because it is more difficult, both chemistry wise and from an enzymatic perspective, to break a CF bond relative to a CH bond, you find many times substitution like this slows down metabolism enough for you to get a half-life within that window of what you're looking for. Okay, so it is these type of metabolic stability studies which kind of point to the fact that one can to point to the fact of understanding where metabolism is occurring and try to modulate that as part of medicinal chemistry. So that kind of becomes like part of your SAR in a sense. Okay. The second thing we are also interested is if metabolism does occur, what are all the metabolites that are formed? Okay. And generally for this, there is no shortcut in a sense. In the olden days, what they used to do is give a dose of drug to an animal, then collect the blood, the urine, the bile, tissues, and then very painstakingly look at all of them to see whether metabolites are present, and if so, which are those metabolites. And typically, this was done using NMR and mass spectrometry. More recently, though, because of developments in mass spectrometry, you find that you can do these studies at a much faster pace and much more easily than it was before. Okay, and what is it that we are looking for? We want to know what is a metabolite, what is the structure metabolite relationship, if at all, and whether a metabolite, if it is formed, is there a chance that it might also have pharmacological activity? And also, you are watching out to make sure that you are not generating toxic metabolites or intermediates which are electrophilic, which can cause what is called a genotoxicity and mutagenicity. Okay, so the aim here is to identify the metabolites. Which are the metabolites? Okay, so here is an example of this type. This is kind of like a model drug. So it is typically will be incubated with liver microsomes and hepatocytes. And after doing the incubation, you might do a very targeted screening for metabolites. What do you mean by targeted screening is sometimes one can look at a structure and knowing what is drug metabolic reactions kind of predict all the possible ways this drug can be metabolized. And then once you know that, you specifically look for those metabolites using your LCMS system. Nowadays, this can be done in a more open fashion using what is called as mass defect filters. So you can just give the compound, set the defect filter, and it will pick out all peaks which are probably metabolite type. And you can then look at them to see which metabolites are formed. Again, as you do this, you're trying to identify what are called as hotspots so that you can kind of feed that information with the medicinal chemistry group so that they can alter the structure and improve the PK of the candidate by altering the metabolism. Okay, so this type of information is part and parcel of the drug discovery process. Then we also look for what are called as differences in the species. Okay, and we do this by cross-species comparison. This is very important because during the drug discovery and development process, by law or due to regulatory requirements, you are supposed to do animal experiments in one rodent species and one non-rodent species. And then this information is looked at by the FDA to kind of get a feel for the overall safety efficacy before the drug will ever be given to a human. Okay, so a lot of pharma companies will have a whole panel of these studies done where you use something like rat, mouse, dog, rabbit, monkey, microsome, or hepatocyte and look at how a chemical entity is being metabolized by all of these systems and which system is the closest to the human. Okay, and the idea is to pick those species as close to humans as possible so that the animal data has much better correlation to what is going to happen later on in a human. So typically it will look somewhat like this. Here is a drug and it's metabolized. This is the kind of like an MS of of the experiment. So you see all of these peaks here are drug derived. Okay. So you can do it in one of two ways. Again, pure MS, or you can also use what are called radioactive drug and look for radioactive metabolites. Okay. So here you can see mouse, there is about 6 or 7 peaks. Rat, there's only like 3 major peaks. The other ones are very minor. Dog has got one major peak and then again 2 minor peaks. This is a monkey, 2 major peaks. Then the remaining 5 are relatively smaller. And here is the human. So you can get a feel that human and kind of mouse seem to be kind of close except for overall quantitative results. And then you start making sure that relative to mouse and rat, mouse seems to be more close, so that will be a rodent species. And then relative to say dog and monkey, monkey seems to be more closer to humans. So I'll use monkey as a non-rodent species. So this is how we make decisions as to which animal models we'll be using when we do our talks and animal PK studies. Okay? Uh, and there's another example here. One of the things we also look for is there any what do you call as disproportionate metabolites? What we mean by disproportional metabolites is when you compare different species, do you find that one species is very, very different than all the others? Or like, for example, human is very, very different from all the others. And if that is the case, the choice of animal system becomes even more difficult. Okay. Here is a drug called momelotinib, and you'll find that when you look at its metabolites, the M21 metabolite is higher in human than both in rat or dog. Okay, so this is a kind of classic example of how you have to be now careful when you look at rat and dog data. Even though they might be close to humans, humans are very different in that one metabolite is very produced in much higher quantities. Okay, and then in terms of wanting to know, this is the drug momelotinib, and the metabolite that we are talking about is this one, whereby metabolism occurs at this position here in the morpholino ring to give you this oxidized metabolite. The rats and dogs don't do this to a significant extent. Okay, so it is information like this that you are after when you are doing this, developmental study. Okay, then we have something called as reaction phenotyping, and reaction phenotyping means that for every metabolite that you have detected, if the metabolite is formed to a significant extent, then you also need to know which enzyme exactly is involved in the production of that metabolite. Okay, here again, you can use pure enzymes or microsomes or supernatants. The idea is in the end, I want to pinpoint which enzyme is responsible for a given metabolic pathway. Okay, so you might find something like this. I'm looking at one metabolite and I'm looking at the different cytochrome P450s, and I find that it is mostly metabolized by 3A4, but 3 other enzymes are also playing kind of not, if you can say, very significant, but not also something which can be ignored. So it appears that 4 enzymes are involved in the metabolism of this drug, and many times this is what you are after. Okay, you kind of avoid drugs which are metabolized with a single enzyme, like say 3A4 only or 2D6 only. You are more happy when you have multiple enzymes doing metabolism. The reason is that in populations you will find what you call a genetic differences, and that you will find people who are defective in one or more of the enzymes. So if that is the case, if all the drug is metabolized by only one enzyme, and that enzyme is absent in a given person, then that person will react very differently to that dose of drug than the entire population. However, if you have 4 or 5 enzymes doing metabolism, then in any person who has got a genetic issue where one enzyme is not working properly, the other 3 enzymes will take over the metabolism, and overall the drug will still be eliminated without a problem. Okay, so reaction phenotyping studies are basically directed towards that, understanding which are those enzymes and making sure the drug is not metabolized only by one enzyme, and if so, that enzyme is not showing variability in a population. Okay, so here is an example of how it can be so significant. Here is the concentration profile of a drug in a group of Chinese subjects, and the 3 groups of Chinese subjects are slightly different from each other. Okay, so if you look at it on the right here, uh, the middle one, okay, which has got a circle here, are Chinese volunteers who are what are called as PMs. PMs means they are poor metabolizers. In other words, they carry a mutation in the gene of an enzyme, making the enzyme kind of inactive. Okay. Then you have another group which are called as, in a sense, extensive metabolizers. Okay. So extensive metabolizers are where they have a very good copy of the gene. It does metabolism very efficiently. And then we have what are called as IMs or intermediate metabolizers who are heterozygotes. That means they have one good copy on the gene which came, say, from the father's side, while the mother's side gene is carrying a defect. So they have half the capacity. Now see what happens when you give a drug to these group of people, and metabolism is mainly because of this enzyme. You suddenly find that these poor metabolizers, the drug levels are higher and they're declaring much more slower than what you see as what is called extensive metabolism. Okay, when you give that drug kind of orally for a long time, you find that at steady state also the concentrations in poor metabolizers are higher than the concentrations in what are called as extensive metabolizers, which is the lowest. So this can be very significant in a population. So this type of information is of relevance when you present the data to the FDA to get permission for use in humans for the first time. Okay. Now, in many cases, if such things are observed in a population, you'll find that the dosage has to be changed when you are using the drug clinically. Okay. So here's an example, something like imipramine. If it is given to an extensive metabolizer, that means person who has got very good metabolic capacity, you typically give it as a dose of 130 or so. But however, if the person is known to be a poor metabolizer, the dose is almost 1/4, kind of 30. So such dosage adjustments, if needed, can be predicted when the drug is being developed, so that later on it doesn't create an issue when the drug is marketed. Okay? The other issue is that nowadays you find rarely do you give drugs alone. You go to a doctor, and typically a doctor always gives you kind of like you can say 4 or 5 drugs which are to be taken at the same time. When that happens, you'll find that there's a potential for one drug to affect the metabolism of the other. Okay, and this can occur in 2 ways. One is that the drug can inhibit the metabolism of another drug which is given at the same time. So now what will happen? 2 drugs are competing for the same enzyme, and the compound which has got a higher affinity for the enzyme will get metabolized faster, while the other one will be metabolized much slower than normal. Okay? Such studies are done again in different types of systems, starting from what are called as expressed enzymes, going all the way to liver slices. And what we are trying to find out is what is the IC50 value for the tendency to inhibit enzymes of drug metabolism, and what is the mechanism of inhibition. Okay, so the experimental design is very easy. You take what is called as a standard drug, which is metabolized by a given enzyme, and do a control experiment to see how much metabolite is formed. Then you repeat the same reaction in the presence of your drug or the drug you are trying to develop, which is shown here in triangle. And if that drug is an inhibitor of this reaction, then you'll find suddenly much less metabolism is seen. Here the metabolites are shown as red dots on green. Here you'll find it is kind of half of in this case. So that indicates that this compound, which is shown in triangle, which is typically your new chemical entity, is an inhibitor of a given reaction. Okay, so you do this study for almost every cytochrome P450 and many of the phase 2 enzymes to make sure that your drug is not going to inhibit other drug metabolic enzymes significantly. And if so, then that will kind of be useful in planning drug-drug interaction studies and then later labeling the compound appropriately when it is presented in the clinic, so that the doctor is able to make sure that he or she doesn't prescribe two drugs which can potentially interact with each other. Okay, so this is information is also generated during discovery and development. In the same manner, just like a compound can inhibit metabolism of another compound, it is well known now that many compounds, when they are given, especially as a part of kind of long duration of therapy, like anti-diabetics or anti-hypertensives, they can actually act as inducers. So when they are given for repeated periods of time, they tend to increase the synthesis of other drug metabolizing enzymes. So what happens is now your body is able to do metabolism much faster than before. So now, because of the new chemical entity that you are administrating, when it's given over a long time and these enzyme levels are increased, then when you give another drug, suddenly you find that drug is metabolized so fast that you get clinical failure. Okay? So it's important that you predict this induction potential of your new chemical entity before it actually enters the market, or to use that information to plan studies to kind of figure out whether it's kind of is this induction significant or is induction non-significant. Okay? And these experiments are also very easy. What you do is you take hepatocytes, you expose them to what is called as just solvent, which is what you mean by no inducer, for say a week or so. And after the exposure, see how much new enzyme has been produced. And that you do by looking for very specific metabolic reactions. Now, this is your control experiments with the solvent. Then you repeat that experiment with a new chemical entity, which might be, say, a CYP inducer. You might find suddenly that when you give it for a long time, now you generate more enzyme. And all these more enzymes are able to do metabolism much faster. So now here, it could be like an induction-based drug interaction, which is at play. So, so you can see all this type of information all kind of filter in, and then for something like valdecoxib, we would know exactly, how quickly or slowly it is metabolized, what are the different metabolites that are formed, which are the major ones, which are the minor ones, what are their structures from NMR and X-ray, and then you give valdecoxib repeated on repeated occasions to confirm whether it inhibits any other enzyme which does metabolism or it induces any other enzyme. Okay, and this then becomes like the drug metabolism packet, which is part of the overall pharmacokinetic packet, which again is a part and parcel of the overall clinical and preclinical picture of a drug. And then this information is evaluated by the FDA, and then they give a go ahead to give it for the first time in humans. And when you give it to humans also, again, you continue with these type of studies to find out which are the metabolites in humans, how quickly or slowly they do, and whether the metabolites are different from what you see in animals in an in vivo setting. Okay, and all this kind of trickles in into the drug discovery process. Now, I've listed only a few of them. Here is a list of a lot of other studies which are also done as part of what is called as ADME studies that support a new drug application. Things like making a radioactive compound and then giving it to an animal and following what happens to that radioactivity, and to kind of make sure you know exactly where the drug is at any given time. Sometimes people also look for biliary excretion studies to see whether a drug is excreted in the bile as is or as a metabolite. Okay. Sometimes this meta-ID is also related to toxicity. So rather than only concentrating on effect, is toxicology also dependent on all of these metabolites. Then on the clinical side, you do things like mass balance and metabolic profile and identification. Again, in a human, We also do what is called as population PK, large scale PK studies to kind of help your dossier become strong, so that the FDA then accepts your information and kind of allows you to kind of give it for the first time in the humans and continue. Okay. Again, here also DD interactions, penetration into breast milk, crossing the placental barrier, going into the reproductive system. All of these things are also studied as part and parcel of the package, which is put in as a PK package for submitting the NDA. Okay. So you can see that the process of just drug discovery and development itself is very complex, but within that, even the drug metabolism part is very, very complex. Okay, because you need to know a lot of things about a given entity. Again, to very quickly reiterate, how quickly or slowly, which are the metabolites which are formed, what is the identity, which are the enzymes which actually do that reaction, is there any enzyme inhibition possible when other drugs are given, is there induction potential that your NC is showing. And when all of this get like what is called a green signal, at least from my ADME perspective, you can give the go ahead for the molecule to kind of go through other tests, which other departments will be doing. Okay. So in a nutshell, I talked, and what I very deliberately didn't talk about, which is a new upcoming field. I shouldn't say new and upcoming, it's already established. That is the role of what are called as drug transporters and how they impact the overall pharmacokinetics of the drug. And you are also familiar with many of these. And nowadays, it's very common knowledge that we know that a lot of proteins are present on cell surfaces, which are responsible for maybe active entry of drug into the cell, or efflux of drug from the inside of the cell to the outside, or movement of drug from one phase of the cell to the other phase of the cell, what we call as basolateral to apical transport of a drug. I have not covered this, but this is nowadays a hot topic in the area of DMPK. Okay, transporters and their role in the overall ADME of a drug. Okay, so hopefully I've been able to give you a nice overview in the 50 minutes or so. That leaves us maybe about 10 minutes to ask any questions that you might have, and I'll be more than happy to answer anything you wish for. Hello. I think it's open. Anyone has any questions? Hello, am I audible?
नहीं यांचा आता क्वेश्चन आन्सर बाकी आहे अजून.
Yes,
Hello.
delegate, if anyone is having any question, you can post it in the comment box, please. I extend my sincere thanks to our expert speaker of session 2 of day 4, Dr. Krishna Iyer sir, who spared time from his busiest schedule to to grace this international e-FDP, e-STP. Today, we had an opportunity to hear your thoughts on the process of drug metabolism. Sir, you explained very deeply the entire metabolism process of drugs, starting right with the basics. We all have clearly understood that this step is the ultimate goal for the drug discovery and development process. This will surely enlighten our future path. Thank you very much, sir. Thank you once again.
Thank you. Thank you.
Thank you, sir. The certificate is shared.
Thank you. Thank you very much. Thank you.
Thank you, sir.
Thank you.